And the next speaker is Juan Domingo Gispert from Barcelonabeta Brain Research Center, and he's going to talk us about their fantastic course and preclinical studies.
Thank you very much for the introduction, and thank you very much to the organizers for inviting the BBRC, the Barcelonabeta Brain Research Center, to present our program, the ALFA cohort. And I will try to do my best to summarize the main results that we've obtained in the last 10 years with this scientific program. As you all well know, Alzheimer's disease biomarkers can be altered many years before the onset of the symptoms. And this is widely regarded as an opportunity to essay preventive treatments. But in order to develop this kind of treatments, we need to understand what might be also the causes that may lead to the accumulation of these abnormal proteins in the brain, like what might be the risk factors contributing to that, and then to test preventive interventions, trying to see whether the modification of these abnormal biomarkers, whether the removal of these abnormal proteins in the brain can break the pathophysiological cascade that ends up with neurodegeneration and cognitive decline. So in order to study and to contribute to all these questions, about 10 years ago, the Pascual Maragall Foundation started the ALFA cohort, the ALFA platform, and started recruiting almost 3,000 cognitively unimpaired individuals, aged 45 to 75 years old. And the common denominator was that all of them, or most of them, were the adult children of Alzheimer's disease patients. So we had this cohort of cognitively healthy individuals that had a somewhat increased risk of developing AD in the future due to the family history. In these participants, we collected their clinical history, lots of information about cognition, genetics, some neuroimaging data, and lots of questionnaires about environmental and lifestyle factors. And that allowed us to select a subset of these participants to create a longitudinal cohort study that would cover all the risk of AD from individuals that would be protected, perhaps because they had protective genetics, to those that were at the highest risk of developing the disease in the early future. And that we selected about 400 of these individuals into what we call the Alpha Plus cohort. And this is a longitudinal cohort study in which we characterize the participants more deeply, and it's a longitudinal study that visits the participants every 3 years. So I will focus first on the results of the first short, the Alpha Mother cohort. These are the main characteristics of the participants. As you can see, they are aged around 55 years. About 2/3 of them are women, and they are fairly well educated. And of course, all of them are cognitively unimpaired. And as we selected them being the adult children of AD patients, we could see that about half of them, the age at where their father or mother were diagnosed were before the age of 75. And about even some of them had 2 of their relatives diagnosed of the disease. As I would be expected with this recruitment strategy, we ended up with a cohort in which we had a higher proportion of participants that carried at least one APOE4 allele, so the highest genetic risk factor for Alzheimer's disease, as compared to the general population. And on the other hand, the protective alleles, the E2 alleles, were underrepresented in our cohort. So we had a cohort enriched for genetic risk factors for Alzheimer's disease. On the other hand, when we look at the cardiovascular risk factors for dementia, we can see that this cohort has a relatively low percentage of participants with high cardiovascular risk profile as compared to some other populations. So we have high genetic risk for AD, low level of cardiovascular risk. Having this high number of APOE4 homozygotes, due to this recruitment strategy, we started focusing on what would be the neuroimaging correlates of these APOE4 homozygotes. We had a very nice cohort of about 70 homozygotes that were cognitively unimpaired. And the first studies, we could see that they already had a decline in gray matter volume in the hippocampus, for example, earlier than the non-carriers or the heterozygotes, as well as a decline in myelin content in the brain. That could be explained because they might be in early preclinical stages. We also looked at cerebrovascular markers in the brain, and we could see that while APOE4 carriers, both heterozygotes and homozygotes, they had a high prevalence of presenting cerebral microbleeds in the brain, the probability did not change that much. So the prevalence is more or less the same. But when the homozygotes present at least one microbleed in the brain, they tend to have more than one. So it looks like their brain is more vulnerable to this early cerebrovascular pathology. And similar results were found when we were looking at white matter hyperintensities. That is supposed to be an earlier and more sensitive marker of early small vessel disease. We could see that the presence of pathological levels of white matter hyperintensities, by pathological we mean FASECAS score of 2 or more, was increased in APOE4 homozygotes, but not in heterozygotes, even after correcting by age, which is a well-known risk factor for white matter hyperintensities, and hypertension, which is again a very well-known risk factor for small vessel disease in the brain. So we have these APOE4 homozygotes that have a more vulnerable brain to early stages of the disease and also cardiovascular disease. And of course, there's something you cannot modify, your genotype. But one thing that these individuals at high genetic risk can do to moderate their risk of developing the disease in the future is to adhere to a healthy lifestyle. It is well known in the literature that these participants with high genetic risk are those that benefit the most from these healthy lifestyles. And in particular, our colleague Aleix Sala, he studied whether self-reported consumption of DHA, which is an omega-3 fatty acid, which is commonly present in salmon or in bluefish, was associated to some protective neuroimaging characteristics in this APOE4 homozygote. And we looked at the AD cortical thickness signature. So it's, we know the regions in the brain that are known to suffer atrophy in Alzheimer's disease, and we make a composite of their thickness. And even in this preclinical population, we could see that higher self-reported consumption of this omega-3 fatty acid was associated to a more protective phenotype. We interpret a higher cortical thickness in the AD signature as these participants having a higher brain reserve as those with a lower cortical thickness in these regions. That is what happened to these APOE4 homozygotes that had a lower intake of DHA. And all this was about APOE4, but of course there are other genetic variants that contribute to Alzheimer's disease. And now very recently, we were also able to receive our results of the GWAS study for the extended cohort, so over 2,500 of these participants in the ALFA plus cohort. And also, this is a very strong test bench to cross that genetic information with all the other data that is collected in the ALFA study. And in particular, all these studies are led by our colleague Natalia Villar-Tejedor. And in particular, we studied that the variability present in a number of polygenic risk scores that are associated to Alzheimer's disease. This is one poster that Patricia Genius is presenting in this conference, so you can see the poster and ask her if you want more details on this study. But we can see, for example, that in our cohort, we have large variability, not only for AD risk, even if we exclude the APOE, gene from the computation of the polygenic risk score, but also from the temporal dementia. So it's interesting because maybe the alpha cohort can also be a good test bench for studying some other neurodegenerative diseases. And for example, when we look at some other of these polygenic risk scores, we can see a high variability of intracranial volume, suggesting that we might have some of our participants with a very high brain reserve and might present cognitive reserve in the future. And when looking at risk factors, we can see high variability in LDL cholesterol levels and also in fat content of the body, also suggesting that we might have a good cohort to show the impact of metabolic interventions in preclinical Alzheimer's disease. And now going to the ALFA Plus study, in which we have also access to the biomarkers of Alzheimer's disease. We didn't have it in the initial study. We can see that these 400 individuals are slightly older. Mean age is 61. Again, about 2/3 of them are females. And here's a high proportion, over 50% of APOE4 carriers. So it's a really biased cohort. It's not representative of the general population, that's for sure. But it's very well enriched to really understand the early pathophysiological events in preclinical Alzheimer's disease. That was our goal right from the beginning, and that is what determined the inclusion criteria in this ALFA Plus study. And it was confirmed when we first had the results of the first biomarkers that were amyloid and tau in the CSF, where we could see that already 1/3 of the participants of the ALFA Plus cohort had abnormal amyloid levels in the CSF and are so to say to be in the Alzheimer's continuum as per the nomenclature in the ATN framework. So we started looking into these AD biomarkers in the ALFA Plus cohort, and one of the first things we tried to work is on methodological approximations to detect very early abnormalities in these biomarkers. And we also had amyloid PET in these participants, and as you may know, when quantifying amyloid in the brain, we simply we make a ratio of the uptake of the tracer in amyloid-avid regions as compared to some reference region. But this kind of ratio, which is called the SUVR, the standard uptake value ratio, is sensitive to the tracer that one is using or how one draws these regions of interest in the target region, etc. So a few years ago, it has been proposed, this centiloid scale, to be able to provide an absolute metric for amyloid accumulation in the brain with PET imaging that states that a centiloid value of 0 is what would be expected on average in young healthy controls, so no amyloid in the brain, whereas a value of 100 is granted to the typical amyloid uptake in AD patients. And having this absolute scale is really interesting because then it allows us to compute thresholds that can be used, for example, to recruit participants into clinical trials or for preventive interventions, like is being used, for example, in the AHEAD preclinical trial that selects participants with centiloids between 20 and 40, or between 40 and between 20 and 40 or over 40. But some years ago, it was not that clear how to establish this kind of centiloid cutoff values. So our idea was to really compute very sensitive and very early values while still retaining some specificity in this very early population. And we compared the centiloid values in our population with the levels of amyloid beta 42 on the CSF, because we know that beta 42 changes earlier in the disease continuum. So as we expected, we have a variation from normal levels of CSF beta, while the PET signal still not changing. So it covers all this area over here. But at some point, the PET signal starts picking up while a beta CSF, the beta is still declining in the CSF, and we have a more kind of linear association. So we computed these centiloid cutoffs in agreement with pre-established cutoffs for positivity in the CSF. And we ended up with a value of 12 centiloids as a very early and sensitive measure to detect amyloid in the brain from PET scans. And this was regarded as a very low cutoff when we reported that. But I'm glad to say that these very similar cutoffs have been validated later on in independent cohorts with Gaussian mixture modeling, so completely different approaches. And that, for example, in some clinical trials, very similar cutoffs are being used now to assess the lack of or the complete reduction of amyloid in the brain in the clinical trial. Centiloids are a scale that can be used in research settings when one can quantify the scans, but in the clinical setting, the most well-established method is to do a visual reading of the scans. And even though the amyloid deposition in the brain is assessed in 4 or 5 different regions in the brain, depending on the tracer, typically the output, the outcome of the visual read is binary. It's either positive or negative, and it's granted positive if only one of these regions is regarded as positive. And it's positive irrespective of there's one or more regions presenting amyloid in the brain. So we thought that this information about whether amyloid was present in one or more regions or in what regions it was present could provide more fine-grained prognostic information for these patients. So the first thing we did is to develop a visual reading staging scale. And in this scale, when if there's no regions, it's negative, of course, but if we have one region in early accumulating regions, like the frontal areas, as we can see here, or in the posterior singular precuneus, that would be stage 1. If you have the two of them, of these two early regions, that would be stage 2. And when other later regions, like the lateral temporal, parietal, striatum, starts also showing amyloid accumulation, that would be stage 3. And again, our idea was that to convey prognostic information for the participants, and this is something we are studying at the moment in a European project called AMIPAD. But very early results from our cohort in Barcelona show that those individuals which are positive by visual read in these late accumulating regions might have some lower cognitive performance in the preclinical Alzheimer's composite for cognition. But we only observe that in the APOE4 carriers. And we still don't know whether this is due because these APOE4 carriers have been having amyloid in these regions for longer, as we know that APOE4 is really promoting amyloid accumulation in the brain, or just because their brains are again more vulnerable to amyloid pathology. So another approach we did in comparing amyloid through PET and CSF is to residualize amyloid PET from CSF. So trying to see which the regional levels of amyloid accumulation in the brain for any given level of amyloid alterations in the CSF, insoluble amyloid. And what we observe is that APOE4 carriers, females and individuals with older age, they have an excess of PET signal for any given level of the alterations in the CSF in amyloid. So all these might be factors promoting amyloid accumulation, amyloid aggregation in fibrillar form in the brain with early soluble amyloid dyshomeostasis. An interesting APOE4 carriers show this excess increment in temporal areas, medial temporal, as we can see very well in this region. We replicated more or less these results with the ADNI cohort, and we could also see accumulation in the hippocampus and in other temporal regions. And again, these are regions of late amyloid accumulation. And we hypothesized that this promotion of aggregation of amyloid in regions of late accumulation might also promote the colocalization with tau pathology in the brain and therefore trigger the neurodegeneration cascade. and promote this neurodegeneration cascading in its participants. And this is something that we will try also to show with tau PET. Aside of these non-modifiable risk factors, we're also studying other modifiable risk factors like pollution, air pollution. And this study, in collaboration with the Barcelona Institute for Global Health, we saw that participants in the ALFA Plus study that were exposed to higher levels of air pollutants, again, show higher levels of centiloid values in the brain, so amyloid accumulation in the brain. But while their levels of amyloid in the CSF remained normal, or at least not associated to these pollutants. And therefore, the conclusion is again that pollution, air pollution, might also be favoring and promoting amyloid aggregation in the brain for any level of soluble amyloid dyshomeostasis. And also having this big cohort of individuals with a familiar history of Alzheimer's disease, we wanted to test whether the proximity to the age of parental onset of the disease was also predictive to amyloid accumulation. In sporadic Alzheimer's, these years of expected onset are not as predictive as in genetic forms of the disease that we saw in the presentation by Raquel Sanchez. But even in these sporadic cases, we could see that proximity to parental age of onset was associated with higher amyloid level, even when corrected by age, sex, ApoE4, and we saw an interaction with sex and with education. Sorry, with sex and age. And this is very interesting because we see that this metric of age of onset is independent of familial history load, so whether they have more or one relatives with the disease, and ApoE4. And this is particularly expressed with women more than men. And this suggests that maybe some heritable factors, which are not ApoE, which might also be contributing to this amyloid accumulation in preclinical stages, particularly in women. Another modifiable risk factor that is associated with Alzheimer's disease is sleep, poor sleep quality. And we have some very early results that link poorer sleep quality, self-reported sleep quality, to some gray matter decreases in areas known to be associated to poor sleep, but also with Alzheimer's disease, like the hippocampus, and also some glucose metabolism decreases in the temporal pole. These are results that we're going to be presenting in the conference in San Diego in 1 month or so. And also we see that the presence of abnormal biomarkers show an interaction in all these kind of effects. But all these are self-reported measures. So we're now setting up a study to have more objective measurements of sleep through actigraphs into 100 participants that will be recruited based on their biomarker profile, and also a subset of 60 of them with polysomnography in collaboration with the sleep unit at the hospital clinic. This is a project, ALFA Sleep, which is led by my colleague Uriol Grau Rivera, who is the lead of the clinical research group at BBRC. And it's, I think, a good example of one independent study with independent hypotheses and objectives, which is nested within the ALFA Plus study, or the broader ALFA program. As I was saying before, now we also have some initial experience with tau PET, and we will also be presenting that in the AAC conference this summer. We had scanned now we have about 60 participants, trying to overrepresent those with already have some biomarker alterations in the CSF. And we have about some participants which are positive. As we expected, the percentage of positivity is not that high. But what I think is more interesting is that as we model the uptake of this ligand for tau across centiloids as a proxy for disease progression, we can see that there is an inflection point very early on, so very around 20 or 30 centiloids, we already see the signal starting picking up. And this is something that, of course, we need to confirm with more cases. But tau PET is really believed to be a more later marker in the AD continuum. And again, that would mean that it might start changing earlier than what is widely believed. Still, all of these changes are in a sub-threshold level. So these are very early and very initial changes in tau. But of course, there's more than amyloid and tau in preclinical Alzheimer's. We know that many other pathophysiological pathways are altered at these stages. And we have a number of biomarkers that we can measure to see whether these systems are implicated, notably inflammation markers, markers of neural injury or synaptic dysfunction, and markers of vascular dysregulation. And in a work that was led by Marc Suarez-Calvet, who is the group leader of the biomarker group at BBRC, we could study some of these biomarkers in the alpha plus cohort. And for example, we could observe that markers of neurodegeneration like NFL were increased in A plus T plus individuals, as well as markers of synaptic dysfunction, astrocytic activation, GFAP, microglial activation, STREM2, again, astroglial reactivity, YKL40, another marker of astrocytic activity, S100B, but not, for example, in IL-6, another marker of neuroinflammation. And the interesting bit of having all these biomarkers that informs us about specific biological pathways is that we can also correlate that with neuroimaging and cognition. And for example, we could see that this component, what I mean a component is that many of these biomarkers tend to vary together. So when one is high, the other one is also high, and one is low, the other one is also low. So we have this component, which is mostly formed by p-tau and neurogranin. Well, these p-tau, t-tau, and neurogranin show a very high correlation in our cohort, but also informed by some other markers of neuroinflammation, like, for example, YKL40, a marker of astrocytic reactivity, but also TREM2. And we could see that this combined tau pathology with neuroinflammation is expressed in A plus T plus individuals, as would be expected because of the tau component, but it's associated maybe in a paradoxical way with increments in gray matter volume and with increments in glucose consumption, particularly in the thalamus in these participants. And even though we don't see a signal, a significant signal with cognition, there is a trend in this component. And it's well known that in preclinical Alzheimer's disease, we have this opposite neuroimaging phenotype. So we have higher gray matter volumes in individuals with positive amyloid markers and even higher glucose consumption. And well, there may be a number of causes for that, maybe just a selection bias. It might be some neuroinflammatory effects in the brain. It may be just compensation effects to early pathology. So this is something that we still need to understand better. And of course, we have now plasma biomarkers, which are really changing the field. And we are also very interested into trying to discover very sensitive plasma biomarkers to reflect early pathology. And in particular, we have been assaying in the alpha plus cohort the p-tau231, which is one of the most promising biomarkers in plasma for Alzheimer's disease. And again, we can see that it reaches the level of abnormality very early on the disease continuum, only at 26 PET centiloids. So again, very, very early. The alpha plus cohort is very good to try to detect these very early events in Alzheimer's disease, and maybe earlier than is typically regarded. This work is under review. We are confident that we can publish it very soon. And you may wonder what happens with p-tau217. And I can tell you it's very close to 231, but it's still a bit later on. So 231 might even be better than 217 to detect early amyloid abnormalities. And regarding looking at plasma and CSF biomarkers, GFAP is very interesting because the plasma GFAP changes earlier in the AD continuum than the CSF counterpart. So we wanted to look at what happened with GFAP as a marker of astrocytic reactivity with glucose PET, with glucose consumption in the brain. about 50% of glucose consumption in the brain is consumed by astrocytes, not by neurons. So we hypothesize that if it would be an astrocytic reactivity to early pathology, we might see a signal on FDG PET. And so when we're looking at plasma versus CSF GFAP, we can see that plasma GFAP really is correlated with CSF beta 40, so it correlates very early with the earliest alterations in amyloid in the CSF. Even in the biomarker negative group, even in that group we see a positive association between GFAP and CSF beta 40 that we don't see in the CSF GFAP. Only we get a signal only in the A plus T plus group. And when we look at correlations with plasma GFAP, we see a positive association between GFAP and glucose consumption in typical AD regions that we would expect to be hypometabolic in AD patients, and also in the thalamus, as we detected in the previous study. So supporting that these paradoxical effects in glucose consumption might be driven by astrocytic activation. But interestingly also, when we look at what happened with CSF GFAP and in interaction with AT stages, we saw that those who are tau positive, so those in the A plus T plus group, the association was reversed and was negative, particularly in these areas that, again, are associated with AD hypometabolism and also regions to show tau pathology. So again, that might be throwing some light on very early pathophysiological events associated with very early Alzheimer's disease. Now, Alpha Plus is finishing the second visit, and we are planning the third one, so this is a longitudinal effort. We are trying to add more and more measurements, but of course, one important thing happened that could break the longitudinality of these measurements, which was COVID. And we also tried to see how COVID could affect our participants, and for example, and the confinement. And for example, we could see that women were more affected in terms of the anxious depressive symptoms, but also, of course, also who were caregivers. Very interestingly, we also showed that amyloid positive individuals had already at baseline higher levels of anxiety symptoms, but they also increased the most during the confinement. And finally, another example of how we study COVID is the work led by Arcadi Navarro, who is our director and the group leader on genetic group, in which we selected individuals at high and low genetic risk for Alzheimer's disease. We obtained some mononuclear cells in peripheral blood and induced pluripotent stem cells to generate brain organoids and infect them with COVID and try to see whether those brains with amyloid and Alzheimer's pathology were more sensitive to the effects of the infection, as measuring a number of biomarkers that we can also look in our participants. Again, an example of how these research infrastructures can be used for more basic research. And it's not only Alpha Plus, we're also starting additional research programs like the BETA-ARC, in which we try to recruit 200 individuals with already some subjective and maybe objective decrease in cognition, and to do a clinical history, neuroimaging, and collect biomarkers to follow them up as they progress. So to end up, I think this is exciting times to do research in Alzheimer's disease. The ALFA study is phenotyping cognitively impaired individuals at the early stages of Alzheimer's disease, and we believe is close to optimal setting to detect these very early pathophysiological alterations and what's the impact on these pathological alterations of risk factors and lifestyle conditions. It allowed us to make several contributions to the development of biomarkers, both imaging and wet biomarkers. And it's also part of European projects, so we are working in collaboration with colleagues to really be able to validate and to extend our findings. I think it's a very good resource for this kind of add-on or nested studies, and we are very happy to listen to your propositions, all data, and samples are available to the scientific community under request. And we believe that the longitudinal characterization of these participants will allow additional insights into the natural history of preclinical Alzheimer's disease. By the way, all of our participants are also eligible for clinical trials, so we're also contributing to efforts in disease prevention. And again, as I show you, we are ongoing another complementary studies to the ALFA plus one. And that's all from my side. Thank you very much for your attention. I would like to thank the ALFA cohort participants, all my colleagues at the Barcelonabeta Brain Research Center, and as well to our funders and collaborators. So thank you very much.
Thank you very much, Juan, for the fantastic presentation. Question? Right.
Thank you, Juan. It was very interesting and very elegant series of studies. Just a question about one of the earliest results you showed. So looking at the effect of ApoE, you were showing that the E4 allele has a reduced hippocampal volume. Now, there's quite a lot of controversy about this because the meta-analysis showed there really is no effect of the ApoE gene on brain structure. We've looked at this in particular because there's a problem to know whether the ApoE gene has an effect per se on hippocampal volume, or whether it just increases your risk of neurodegenerative process, and once that starts, your hippocampus shrinks. So did you look longitudinally at that result and see whether the ones that, your E4s, that actually showed a smaller hippocampal volume, it was just that they were already on the road towards neurodegeneration, and it's not the gene itself?
If I understood correctly, you're talking about the lower levels of gray matter volume in the hippocampus in APOE4 homozygotes, and whether that is a pure genetic effect or whether it's due to the pathology. No, I think it's due to the pathology. We know from developmental studies that APOE4 carriers have larger volumes in middle temporal regions, including the hippocampus. So they might have a faster or better development, but that also makes them more vulnerable to early stages of the disease. So I really believe that the alterations we find in gray matter volume in these APOE4 homozygotes, they have different causes. We also found some increased volumes, so that we might have some neuroinflammatory effects, for example. We might have simply some effects that are just the genetic effect that has been there forever and do not interact with pathology. But specifically about these hippocampal decrements, I think this is due to early Alzheimer's disease. By the age that we're seeing these reductions, fairly majority of these participants should have amyloid in their brains, and maybe,
And just one other question, if I can. The one also was very interesting, showing increased grey matter in biomarker-positive individuals. They were in some pretty unusual places as well. So primary visual cortex seemed to be one of the areas. Any explanation for that?
Not really. These higher gray matter volume in individuals with abnormal Alzheimer's biomarkers is kind of a robust finding. It's been found for the last 10 years in several independent cohorts. As I was saying, there may be a number of causes for that, including a selection bias on how we select these individuals. So we really need longitudinal studies, which are really few of them, and we are really looking forward to be able to contribute to that. But more specific to your question, I can't explain the signal in the visual cortex, in occipital areas per se. There are a relatively small percentage of individuals with amyloid that have a accumulation of amyloid in the occipital cortex. It's about 15% of them in cognitively impaired populations. But I don't really expect that to be the reason why the presence of AD pathology is determining this higher gray matter volume in the occipital areas. I don't know.
Okay. Well, congratulations, excellent work.
Thank you.
You report that you don't see a correlation between GFAP and the CSF with other AD biomarkers, but you do see a correlation between plasma, GFAP, and the biomarkers. Am I right? That you see a correlation between GFAP in plasma and AD biomarkers, but not CSF GFAP. Is that correct?
We see a very tight correlation between GFAP in plasma and beta-42 in CSF.
But not with GFAP in CSF?
With GFAP in the CSF, this correlation is much worse and only starts showing a signal only in those individuals who are also tau positive. So we will have a correlation between amyloid beta and CSF GFAP in tau positive individuals.
And do you see a correlation between CSF and plasma GFAP?
Only in those who are not tau positive. So I could show you my slide if I can come back to it, but we see a very good correlation between CSF beta-42 over 40 and plasma GFAP. That was the best correlation, independent of AT status. That correlated very well. Then the correlation between, when we start looking at CSF GFAP, the correlations were piecewise different, so there were interactions depending on the AT status. And the tau positive group was the one that behaved different as the tau negative groups.
Thank you.
I think we have no more time for questions. I would like to thank both speakers for the outstanding presentation and for keeping on time. Thank you very much.